Over a span of just 5 weeks, an outbreak in the western Indian state of Gujarat claimed the lives of 25 people. This sudden surge in mortality echoes a similarly brutal spike from 2024. The victims share a tragic demographic profile. Almost all of them are children under the age of 15, living in rural or semi-rural environments. The pathogen responsible for these fatalities is the Chandipura vesiculovirus, commonly known as CHPV. It causes acute encephalitis syndrome, leading to rapid and severe inflammation of the brain. Historically, CHPV carries a staggering fatality rate, killing between 56 to 75% of the patients it infects. A disease that can rip through a community and kill more than half of its pediatric victims in a matter of days commands the highest level of public health urgency. Public health officials from the National Joint Outbreak Response Team have flooded the affected regions to contain the spread, but they are hitting a wall in the field. To stop a vector-borne disease, scientists have to find the insects carrying it. Teams have collected and frozen thousands of potential carriers, including sandflies, mosquitoes, and ticks, from the homes of the victims. In the lab, technicians crush these insects, extract the nucleic acids, and run RT-PCR tests to amplify any trace of the virus. Yet, despite the massive scale of the outbreak, every single insect tested has returned negative. The geography of the infections is also confusing researchers. Sandflies, the historical prime suspects, are highly endemic to eastern Gujarat, but current human cases are emerging far to the west, in regions like Saurashtra. Without a localized host to target, officials cannot execute targeted extermination campaigns. The complete inability to find the virus in local insect populations turns a predictable seasonal outbreak into an untraceable phantom threat. This virus first emerged in the medical record back in 1965. Scientists isolated it from 2 patients suffering from a febrile illness in Chandipura, a small village in Maharashtra. This map illustrates the historical footprint of the pathogen. The red clusters represent the endemic zones, showing how the virus maintains a grip across central, western, and southern India. These outbreaks follow a strict seasonal calendar. They spike almost exclusively during the early monsoon season, when heavy rains create stagnant pools of water, allowing insect populations to rapidly multiply. The historically established primary vector for this virus is the phlebotomine sandfly, specifically the female species that feed on blood. While laboratory studies show that mosquitoes are perfectly capable of transmitting the disease, wild isolates and historical evidence continually point back to the sandfly as the real-world culprit. The virus relies on a highly specific biological alignment. It requires the right geographic reservoir, the seasonal trigger of the monsoon rains, and an explosion of specific insect vectors to reach its human targets. Looking at this diagram, we see the rod-shaped structure of the Chandipura virus. It's an RNA virus in the Rhabdoviridae family, related to rabies. Infection begins the moment a vector bites a host, depositing these internal viral RNA strands directly into the bloodstream. Unlike respiratory viruses, Chandipura is strictly neurotropic. It rapidly breaches the blood-brain barrier, invading the central nervous system. Inside the neurons, viral replication triggers extreme cellular stress, releasing reactive oxygen species that destroy surrounding tissue. The body responds with a flood of immune cells, creating a massive hyperinflammatory cytokine storm. For a child, the clinical manifestation of this brain swelling is terrifying. It begins with a high fever and severe bouts of vomiting, escalating rapidly into convulsions, an altered sensorium, and finally, a coma. The biological clock on this disease is incredibly short. This entire cascade, from the first fever to severe neurological deterioration, happens in just 24 to 48 hours. The virus effectively weaponizes the host's own biology. By provoking such an extreme immune response, the child's own defense mechanism actually becomes the primary cause of fatal brain destruction. Despite scientists knowing about Chandipura for nearly 60 years, there is currently no approved vaccine to prevent the infection. There is also no specific antiviral treatment capable of curing the disease once it takes hold. Patient survival relies entirely on early detection and immediate intensive supportive care in a hospital setting, stepping in before the brain damage becomes irreversible. Because medical interventions are strictly supportive, communities have to rely on aggressive vector control countermeasures. This includes indoor residual spraying and fogging with insecticides to kill the insects where they breed. Families are instructed to fill cracks and crevices in their mud walls to eliminate breeding sites, and to protect sleeping children with insecticide-treated fine mesh bed nets. In the absolute absence of a biological cure, mechanical prevention at the community level serves as the only viable defense against the virus. The current public health strategy has a critical flaw. Medical teams are largely collecting and testing insects only after an outbreak has already started and children are dying. Experts argue that stopping this pathogen requires deep, year-round entomological surveillance across all endemic hotspots, regardless of whether there's an active outbreak. Researchers also need to identify the unknown animal reservoirs, the domestic or wild hosts that carry and maintain the virus quietly between human epidemics. Defeating the Chandipura virus requires outsmarting it in the dry season. We have to find where it hides, before the monsoon rains unleash it once again.